So muscle and movement. Further information, you can look at muscular system and muscle contraction. So muscular system and muscle contraction. Individual Wikipedia articles. So in vertebrates, the muscular system consists of skeletal, smooth, and cardiac muscles. It permits movement of the body, maintains posture, and circulates blood throughout the body. Together with a skeletal system, it forms the musculoskeletal system, which is responsible for the movement of vertebrate animals. MES notes, skeletal muscles, commonly referred to as muscles, are organs of the vertebrate muscular system that are mostly attached by tendons to bones of the skeleton. The muscle cells of skeletal muscles are much longer than in the other types of muscle tissue and are often known as muscle fibers. The muscle tissue of a skeletal muscle is striated, which is having a striped appearance due to the arrangement of the sarcomeres. Let's see this.
Sarcomeres.
Yes, sarcomeres, which are the smallest repeating striated muscle tissue. Skeletal muscles are voluntary muscles under the control of the somatic nervous system. We'll get to that in a bit. So here is a schematic here, skeletal muscle setup here. So if you look at this runner right there, zoom in, so you have bone, and you have a tendon which attaches the muscle right to the bone, and then this termed epimysium. There's the vesicle, and then there's the muscle fiber inside. There's blood vessels. There's a perimysium set up inside there. So look how it's made. It's pretty ridiculous. It's like bundles of fibers. And there's the muscle fiber single cell. You zoom in this one, and then it becomes this. So you zoom in this. Now you have another bundle inside bundle. So this is some super strong. It's super strong muscles right here, or just a super strong connective setup here, and basically striated organs right here. So muscle fiber, single cell, multinuclear right here. Yeah, so they have multinuclei, multiple nucleus inside each cell, which is quite fascinating. So these dots there. If you zoom out, you have a microfill now. So here is one sarcomere. And this is a myofill, this segment. Now you have this is a myofibril inside there, the full length. And now you have one sarcomere as a segment over there. Remember what the sarcomere was? That is the smallest repeating striated muscle tissue. So this is the smallest repeating striated tissue. It's going to keep repeating on and on. So here's one sarcomere, and if you zoom into it, what you get is actin, myosin filament, and titin right here. And it just keeps repeating on and on. Now, if you zoom into this part right here, yeah, so you keep zooming in, you're going to get a lot of interesting stuff. So you get here thick myosin filament over here. Then you have the hinge stalk, myosin head. And then this is a myosin/actin cross bridge. And this is... this is in between here, but then look at these cases right here, the actin. So you have this set up. When you zoom this part out, you get this, and this part in here, you get this part in between. All right, so you have, this molecules are called troponin. You have tropomyosin. There's the actin, these big circles there. And then you have thin actin filaments around it. So thin actin filaments around, and there's the actin inside there. All right. So let's zoom out and try to make sense of some of this. All right. All right, so we're zoomed out. Yes, basically you take a fiber tendon connected to a bunch of these long cells or these long tissue right there. And then zoom inside, there's bundles inside. And then inside that bundle, inside this bundle, this muscle fiber also has muscle fiber single cells inside. Yeah, so the bigger bundle is this vesicle, and then inside this bigger giant one called the epimysium, and then you have this super small one here, the muscle fiber. So this is some super strong tissue and organ. So a sarcomere, from Greek flesh, or this right here, sarx, and meros, meaning flesh part. So yeah, flesh and part is the smallest functional and repeating unit of striated muscle tissue. And again, recall striated is just appearing like stripes. So having striated, having a striped appearance due to the arrangement of the sarcomeres. So it's all arranged as just in these bundles like that as stripes. So it's all going parallel to each other. And yes, the actin is protein, etc. I'm not going to go over too much of the individual parts there, but just to show you how this looks like there. And also the next part I want to go over here. So so basically these skeletal muscles are under the control of the somatic nervous system. And let's take a look at the somatic. So the somatic nervous system, or SNS, or voluntary nervous system, is the part of the peripheral or the outside or extending to the body nervous system associated with the voluntary control of body movements via skeletal muscles. Yeah, so basically think of the ones where you're voluntarily controlling your muscles. For example, if you flex or pick up something, you're activating your muscles to basically contract and lift, etc. So smooth muscle is an involuntary, non-striated muscle, so called because it has no sarcomeres and therefore no striations. Yeah, so this is in contrast to this right here, the skeletal muscles. Skeletal muscles is one connected. That's the striated ones. It's connected to it. But now if you have smooth, and then you have the cardiac, the heart ones. We'll just go over how this looks like. So smooth muscle is an involuntary, non-striated muscle, and has no sarcomeres and therefore no striations. So it's not striated. And here's a zoom in like this. Zoom in of it. So smooth muscle shown in the tunica media layers in the walls of arteries and veins. So you have these smooth ones get bend, so it's not as rigid as the skeletal muscles. So there's the lumen, the opening. There's the valve there. This tunica intima, or the endothelium cells. Then there's the tunica media, smooth muscle and elastic fibers around there. And tunica externa, connective tissues and elastic fibers. There's artery and there's vein. So you have the smooth muscles in the tunica media, these layers in the walls of arteries and veins. So you have these smooth, non-striated setups over there. Then non-striated, and look how it's like a whole bunch of stuff all together. And so it's non-striated, not in a stripe pattern like this. All right, and now the cardiac muscle, also called heart muscle or myocardium, is one of the three types of vertebrate muscle tissues, with the other two being skeletal muscle and smooth muscle. It is involuntary, striated muscle. So notice the combination. This one is involuntary, non-striated. The skeletal is voluntary, striated. And this one is involuntary. Cardiac muscle is involuntary, striated. That constitutes the main tissue of the wall of the heart. The cardiac muscle, myocardium, forms a thick middle layer between the outer layers of the heart wall. That's the heart wall, the pericardium. Yeah, so myocardium is the heart muscle, and there's the heart wall, the pericardium. And the inner wall, so the cardiac forms a thick middle layer. So we should bold that here. It's between the outer layer of the heart wall, called the pericardium, and the inner layer called the endocardium, with blood supplied via the coronary circulation. And coronary circulation is the circulation of blood in the blood vessels that supply the heart muscle, the myocardium. Yeah, because the heart also needs blood to function, all of its muscles and so on. Coronary arteries supply oxygenated blood to the heart muscle. And cardiac veins then drain away the blood after it has been deoxygenated, because the rest of the body, and most especially the brain, needs a steady supply of oxygenated blood that is free of all but the slightest interruptions. The heart is required to function continuously. Yeah, so the heart needs to keep going forever and non-stop, because the whole body, and especially the brain, needs constant oxygen and then continuous functioning. Therefore, its circulation is of major importance, not only to its own tissues, but to the entire body, and even the level of consciousness of the brain from moment to moment. Yeah, because if you just hold your breath for longer than a few seconds, you'll realize you're going to need that oxygen. So interruptions of coronary circulation quickly cause heart attacks, myocardial infarctions, in which the heart muscle is damaged by oxygen starvation. All right, let's take a look at some of these veins right here. So coronary arteries labeled in red text, and the other landmarks in blue text. So what we have is, so here's the coronary arteries. It's connected. So this is right atrium. There's the left. Then this is left atrium somewhere over here. So anyways, this is right atrium. Notice the connection here is the aorta, which heart pumps out, but it also connects right here into the right coronary artery, RCA. Yeah, so it's even though there's the main artery, main aorta going out to the body, there's still that extra vessel to go and pump blood to the heart. And there's posterior descending artery, PDA, and then there's right marginal artery, RMA. So everything is everything's labeled. There's a right ventricle over here, and there's a superior cava, vena cava, and then there's the left pulmonary artery, and this is pushing out here, and then the other side is on the other side. And what we have is, then there's the veins coming to the left pulmonary veins coming back inside there. And the coronary arteries are in red. So you have the left coronary artery right here. That's this one right here. So this is the right one, this is the left one, to get both atriums and ventricles. And then what you have is this over here is the left anterior descending LAD, or intraventricular artery. This setup right here is the left or circumflex artery, then M1, M2 arteries there, then D1, D2 diagonal branches. And this one's looks like on the backside there. That's the M1, M2 left marginal ones on the backside. And there's the left ventricle here. Yes, very, very fascinating stuff here. And the main ones are the right and left coronary arteries. So the RCA and LCA. Anyways, that's some interesting stuff there. All right, let's continue further. And this, let's compare the different types of muscles right here, and we could zoom out now. All right, so now let's take a look at this figure. This is types of muscle. So we have this is from freepic.com, and let's take a look at how this looks like here. So what we have is the muscle ones connected to the bone. The skeletal muscle ones, that's the striated muscle cells. That's the skeletal muscle connected to the bone. So it's going to be super strong and it's super all in one direction like that, like stripes. And then you have the cardiac muscles. It's going to look something like this. It's striated, but it's not as thick like this one. So you have some openings over there because it needs to be more flexible. Then you have the smooth cells here. So it's not striated, but it needs to be very flexible for the different various organs, blood vessels, and so on. So yeah, smooth muscle cells, etc. Very interesting. And I also note this one here is voluntary. This is involuntary. This is involuntary, but this is striated. This is non-striated. These are still both striated like that. All right, let's continue. So skeletal muscle contractions are neurogenic as opposed to the heart being myogenic. As myogenic, that's the rhythmic beating there. As they requires rhythmic that does not require neural input. It just keeps beating from the muscle, just the muscle contractions. As they require, neurogenic muscle contractions, as they require synaptic input from motor neurons. A single motor neuron is able to innervate or activate, distribute or to stimulate multiple muscle fibers, thereby causing the fibers to contract at the same time. Such as flexing your biceps. Once innervated, the protein filaments within each skeletal muscle fiber slide past each other to produce a contraction, which is explained by the sliding filament theory. The contraction produced can be described as a twitch, summation, or tendinous, or tetanus.
Tetanus muscle spasm.
Tetanus or muscle spasm, depending on the frequency of action potential. So you get twitch, summation, or tetanus, or muscle spasm. Unlike skeletal muscles, contractions of smooth and cardiac muscles are myogenic. So skeletal muscles are neurogenic, so basically voluntary. And then unlike skeletal muscles, contractions of smooth and cardiac are myogenic, rhythmic, involuntary, etc., as they are initiated by the smooth or hard muscle cells themselves instead of a motor neuron, so instead of your brain firing neurons to activate. Nevertheless, the strength of their contractions can be modulated by input from the autonomic nervous system. The mechanisms of contraction are similar in all three muscle tissues. Yes, contraction is similar, but the main difference here, you have the myogenic versus smooth and cardiac versus the neurogenic skeletal muscles. But still, the smooth muscle and the cardiac can still be modulated or affected by the autonomic nervous system, such as just basically the stuff that goes automatically. So if you're scared, etc., or running a lot, you're gonna obviously start heart's gonna beat harder or faster. Via this autonomic nervous system, giving it extra signals. So MES note a motor neuron or motoneuron or efferent neuron. See the pronunciation here.
Efferent.
Yeah, so efferent. Yeah, so this efferent neuron or motor neuron is a neuron whose cell body is located in the motor cortex, brainstem, or the spinal cord, and whose axon or fiber projects to the spinal cord or outside of the spinal cord to directly or indirectly control effector organs, mainly muscles and glands. So basically the motor neuron, the cell body is inside the motor cortex, or basically the brainstem or spinal cord, and the axon is projecting outwards to the spinal cord or outside the spinal cord to directly and indirectly control the organs, such as muscle cells, glands. The motor cortex is the region of the brain involved in the planning, control, and execution of voluntary movements. There's a YouTube video here that illustrates this setup. Yeah, we'll illustrate this figure right here, and we'll get back to this in a bit. And here's added some screenshots from that video, and let's go over that a bit slowly just to grasp the filament theory of contraction. So muscle contraction 3D video. So let's copy and go there. All right, so here's what we get when we teleport here. So muscle contraction 3D. This video shows the contraction of skeletal muscle fibers and the molecular events involved. Let's just zoom in like this. All right, just zoom in a bit more. All right, so that should be good. And let's play from here. Okay, so you have the striated muscle cell contraction. So there's a muscle.
Striated muscle is made up of bundles of multinucleated muscle cells.
All right, so yeah, muscle made of bundles of nucleated muscle cells inside there. So there's the myofiber bundle
called myofibers.
And then the myofiber, each one has these myofiber bundles, and then you have this myofiber inside there.
A myofiber is packed with myofibrils.
All right, so myofiber has myofibrils, so it's even more compact.
That extend the length of the cell. A sarcomere, the functional instructional unit of muscle.
All right, so there's a sarcomere, the repeating part right there. The green is actin, myosin is the red right there, and there's the Z-disc, the end of the sarcomere.
Consists of thick myosin filaments and thin actin filaments.
Then here's a zooming in through it.
Titin keeps thick filaments centered during...
Yeah, so that's titin, keeps those centered. Yeah, so keeps the filament, the myosin right there. So notice the titin's in blue, and there's the filament, the myosin around it.
During contraction. In muscle contraction, filaments centered during Titin keeps thick filaments centered during contraction. In muscle contraction, actin filaments slide past myosin filaments.
Yeah, so the actin filaments slide past myosin filaments. Notice how this green one slides to this one goes from the left side to the right, and this one goes from the right to the left. Let's take a look at that again. And
in muscle contraction, actin...
Yeah, so it pulls that. Notice how it's pulling it in. Yeah, or at least the appearance of pulling in. It could be just a reaction that pushes it open.
In muscle contraction, actin filaments slide past myosin filaments.
Yeah, so they slide past it. So once it touches here, this slides over.
Neither filaments change in length, yet the sarcomere becomes shorter. The force required for this movement is generated by myosin heads.
Okay, so the force is generated by the myosin heads there. So there's the myosin filament there. It connects. There's the titin that keeps the myosin centered. And so let's see the other terms. This is the this is the tropomyosin right here. This yellow there. Notice how it's spiraling around. And yeah, we'll keep going.
Which make cross-bridge attachments to actin. Myosin heads walk along an actin filament, using ATP for energy. Tropomyosin is part of the complex that prevents myosin from sliding along the thin filaments.
All right, so this is the tropomyosin. So it prevents the myosin from sliding along. Notice how it's curved over there. So basically, because of this curved setup, instead of it keep sliding, this will be a connection that stops it. So basically, this goes and stops it here. Yeah, so these are thin filaments, but then it prevents it just adds another barrier there. This as part of the overall complex to prevent sliding of this myosin all the way across. All right, let's keep going.
A myosin head binds ATP.
Yeah, so myosin binds ATP. Let's see that again. Was it ATP or ADP?
Myosin head binds ATP, and as a result,
binds ATP.
Detaches from the actin filament.
Yeah, so then when it binds with ATP, it removes from the actin filament.
It rebinds in a new position.
Yeah, so it then rebinds over here.
Phosphate release.
The phosphate release, now you get ADP.
Triggers a power stroke that moves the actin filament.
Yeah, okay. Yeah, let's go back here. So basically, when this phosphate's released, now the ATP, the adenosine triphosphate, becomes an ADP, adenosine diphosphate, and then that connects over here, and that initiates the power stroke. Let's see how this looks.
Release triggers a power stroke that moves the actin filament.
Yeah, so then going back here. So then this myosin drags it over to the other one, drags it over to this side here. So this actin moves over to the right.
ADP dissociates,
and then once after that, ADP dissociates, and then it needs another ADP to release.
The process is repeated.
No, then the other one goes over there, connects here, release phosphate, power stroke, drags it over, and this one gets slide over there. So this drags it over across here. It's very, very fascinating. All right, hopefully you grasp all that. Let's take a look now. All right, we're back here, and let's just scroll up back to the screenshots and just to illustrate that video. So what we have is there's a sarcomere, there's a muscle fiber or cells, considered a cell, and then basically has a whole bunch of multi-nucleus inside there, so multi-nuclei all the way inside. It's basically considering this whole thing as a cell. And then now inside here you have a microfibril. Yeah, so inside is this part of that bundle there. You're gonna have microfibers, or this is the muscle fiber in this case. Part of a bigger bundle, and then this goes over here, muscle fibril, and then you get all the way over here, Z line, there's a sarcomere, and there's a Z line end there. So you have a sarcomere here, zoom in, and basically theorize, apply theory. You can have a titin, this other protein filament here that goes in the center, and this makes sure that the myosin moves in the center there. Yeah, during contraction, it's always going to be aligned in the center. And there's the actin, this green stuff there, and then the tropomyosin in that yellow, and notice how it's spiraled around there, so it prevents sliding. So yes, because now it has a spiral, so instead of going straight through that actin, you're going to have this little barrier there. So anyways, so that's how it looks like. This is that disc, the endpoints. And let's have a look here. And notice this one is, let's just make it a bit smaller so that you can see the contraction. All right. So this one, in this case, they have, in this case, it's one at a time. They're showing the myosin heads one at a time, dragging the actin, but yeah, it could be basically at the same time, depending on if it's getting the ATP, etc. at the same time. So anyway, just make this smaller so we can see the difference here. And yeah, so notice how it's out here. So this one's sticking out once it connects, and then it drags it in. And this is going to be slightly shorter. Yeah, so you see this setup right here. So you see that gap, and then the next one, once it pulls it in, you'll have barely any gap there. And that's actually after a couple strokes here. Yeah, so after a couple strokes, you'll get it all the way down and contract there. And this one prevents further contraction, so you can't keep contracting your muscles into absolutely tiny. So anyways. Yeah, so here's the zooming in setup there. So there's the titin keeps the myosin centered, and there's the actin is the green, and it's in spiral. This tropomyosin in between prevents sliding. So now this is trying to go straight, so otherwise it would have to turn sideways to get around it. So then this prevents it, etc. So it prevents further sliding. So it just only goes here and connects it, etc. Keeps going on and on. And there's the walking setup. And yeah, so here this one reaches out. and pulls this to the right. So you can see that here. This one pulls it, and then it's going to go over here. So that pulls it. Yeah, very, very fascinating. All right, so if we look at this setup here, let's make this a bit smaller. It's from that same video, and let's make this smaller. So this one, ATP goes in here, dissociates it, or energizes it. Energizes myosin by getting the ATP, the energy currency, and then this releases it. And let's take a look at that video again, just near the end part right here. So just read that up. This filament theory is a bit detailed or a bit complicated. All right, let's just go back here. So what happens here is it absorbs the ATP.
It binds ATP.
This part right here is released ADP, so this one binds ATP,
and as a result, detaches from the actin.
So when it binds it, it detaches, and then it energizes it, and it gets released. Basically, this energizes it can get off of it.
Filament. It rebinds in a new position.
And then it rebinds a new position after that energization.
Phosphate.
And then phosphate's released, and then there's a power stroke. And
release triggers a power stroke that moves the actin filament. ADP.
And then ADP dissociates.
And the process.
And then you need a new ATP to energize it to move it. Okay, so now it makes actual some sense. So ATP goes in here, binds with this, energizes it. This jumps over and binds to another spot. All right, let's just erase this. All right, yeah, it binds to another spot, and then what ends up happening? Yeah, this jumps over. It's connects to another spot. Connects to another spot, and then the ATP releases a phosphate, and then that triggers the power stroke and moves the whole actin to the right there. So it drags it over, and then the ADP dissociates. Yeah, so then the ADP dissociates and repeats. So then this one is on this case dissociates and then repeats. So this is when you need a new ATP inside it. So yes, very, very interesting setup here. All right, so now let's take a look at this figure now. So this is from the fitnesstraineracademy.org. So the sliding filament theory, and I'll do a note. There's a note: the right image should have the myosin connection further to the right end. Yeah, I believe. So anyway, so number one, myosin heads split ATP and become reoriented and energized. All right, so the ATP comes in this case it says it's splits ATP. So there's ATP energized. It splits it and becomes reorientated. So it's over here. Then it's gonna move outwards. It's gonna attach to here. So it's gonna reorient itself, move it over here, and now it has an ADP and P over there. So yeah, it does split the ATP. So now it's together there. And then number two, myosin heads bind to actin again, forming cross bridges. I believe that's just a bridge here. So there's two bridges over there. But then this one here should be to the right. It should be somewhere here actually. Just in this illustration here. I believe it should be here. So let's erase that. Yeah, so erase that. So it should be here. So note the right image should have the myosin connection further to the right. That is what I think. So it should be connected here, and then myosin heads rotate towards center, and there's a phosphate release as well. So phosphates gets released, and then it rotates to the center. In other words, it's moving over to the left. So notice how this position's right there. So this is going to be dragging it over, and that's why this spot is over here. So myosin heads rotate towards center of the sarcomere. In other words, power stroke. So it's a power stroke over there. But this connection should be right here. And then the next one, as myosin heads bind ATP, the cross bridges detach from actin. Yeah, so then this is connected there. The ADP dissociates as well, and then you'll need a new ATP. So as myosin heads bind ATP again, the cross bridges detach from it. So then more ATP, another one connects over here. You're gonna release it like this, and then reorient it into another position. And this would relax, so it should go to the right more. Grab somewhere here and drag it, or grab somewhere here and drag it. Yeah, it keeps dragging. That's fascinating. Let's just erase all this drawings. All right. So just erase all that. Then let's continue further. Now we're dealing with action potential, and we mentioned it earlier. Yeah, and here if I just scroll all the way back up here, so the contraction produced can be described as a twitch, summation, or tendinous muscle spasm, depending on the frequency of action potentials. And this is that contraction of the skeletal muscles. Yeah, so that was dealing with skeletal muscle contractions. And so those are the action potentials that generate all this setup over here. So going over, so in physiology, an action potential occurs when the membrane potential of a specific cell location rapidly rises and falls. This depolarization then causes adjacent locations to similarly depolarize. Action potentials occur in several types of animal cells called excitable cells, which include neurons, muscle cells, endocrine cells, and in some plants. So the action potential is set up. Here's an illustration of it. So as an action potential, nerve impulse travels down an axon, long, slender, conducting projection of the nerve cell. There is a change in polarity across the membrane of the axon. Alright, so here's this video here. So basically, action potential is a changing polarity. So let's say if you have all plus on one side, this is the nerve. So this is the nerve right here, or the nerve, the main cell area, and then you have the axon is this long, slender path right there. It's the axon. And then what you have, so you'll have a polarity here, a default one, plus on one side, minus on one side. And then when you have an action potential, the nerve, what it does is flip it one side. And then that will drag all the way across. So basically it's like a wave, an electric current wave. So once you get a plus here, then they want to alternate. So plus always wants to, the negative wants to go back to this side here. And basically set up like this. The exact details, that's for further video. Yeah, and if we scroll back up here, so the skeletal muscles, they are, basically they'll need a single motor neuron is able to innervate or stimulate muscle fiber cells, thereby causing the fibers to contract at the same time. So basically, the skeletal muscles require the input from the action potential, so you get the electric signal and then activates it, and then the filament kicks in there, and the myosin grabs the ATP, etc., and it keeps contracting. So the mechanism is through the ATP, but then the action potential is required to stimulate the muscle cells. Yeah, which involves grabbing the ATP. So, anyways, continue further. So an axon or nerve fiber is a long, slender projection of a nerve cell or neuron in vertebrates that typically conducts electric impulses known as action potentials away from the nerve cell body. So there's a nerve cell body, there's the axon, all the way across there and connecting to the organ that you're affecting, such as muscle cells or skeletal muscle cells. In biology, depolarization, this polarization, we mentioned it here. So this is the action potential when the membrane of potential of a specific cell rapidly rises and falls. So this depolarization then causes adjacent locations to similarly depolarize. Yeah, so let's go over this depolarization. So in biology, depolarization, or British English depolarization with a S instead of a Z, is a change within a cell during which a cell undergoes a shift in electric charge distribution, resulting in less negative charge inside the cell compared to the outside. And so less inside than outside. And depolarization is essential to the function of many cells and communication between cells and the overall physiology of an organism. So most cells in higher organisms maintain an internal environment that is negatively charged relative to the cell's exterior. Yeah, so that's interesting. So notice how inside is negatively charged, and then once you flip it, this becomes a plus, and now this plus will keep dragging across there as a plus wants to. As a plus wants to basically go back to the outside as well as move to the right as it's just being pushed over there. You can view it as basically water being pushed, or a water wave, etc., or something like that. So anyways, this difference going over here. So yes, most cells in higher organisms, like animals, etc., maintain an internal environment that is negatively charged relative to the cell's exterior. This difference in charge is called the cell's membrane potential. In the process of depolarization, the negative internal charge of the cell temporarily becomes positive, less negative. This shift from a negative to a more positive membrane potential occurs during several processes, including an action potential. During an action potential, the depolarization is so large that the potential difference across the cell membrane briefly reverses polarity. And then with the inside of the cell becoming positively charged, and then it will need to switch up, and you can have that full action potential. The change in charge typically occurs due to an influx of sodium ions into a cell. So you have sodium ions into the cell, it was positively charged. Although it can be mediated by an influx of any kind of cation, let's see the pronunciation, how this is pronounced.
cation,
Yeah, cation or efflux, escape of any kind of anion. So you could have the negative charge getting out or the positive charge getting in. And let's take a look at this. So action potential in neuron showing depolarization in which the cell's internal charge becomes less negative or more positive, and repolarization where the internal charge returns to a more negative value. So here's membrane potential in millivolts. So initially it's negative, so you want a negative charge, and then it goes up depolarization, so it's becoming positive, and then you'll have the repolarization set up here. And in action potential, so you'll have this can be depolarization becomes positive, and then it wants to go back down to its normal one. So you have repolarization, and then you have a segment here, hyperpolarization, where it's lower than it's lower than this one here, so it shoots past its normal setup, and then it'll then eventually get back to normal over there. And hyperpolarization is a change in the cell's membrane potential that makes it more negative. It is the opposite of depolarization. It inhibits action potentials by increasing the stimulus required to move the membrane potential to the action potential threshold. And I believe this is the threshold in this case, this line there. So it will, basically lower here. You're going to need more energy, or more stimulus, more ions, etc., more cations, or remove more anions to get it over to the threshold where it can depolarize and drag over the full action potential. Just very, very interesting stuff. Anyways, let's continue further. So in invertebrates such as worms and earthworms and leeches, circular and longitudinal muscle cells form the body wall of these animals and are responsible for their movement. Yeah, so it's circular and longitudinal. In an earthworm that is moving through a soil, for example, contractions of circular and longitudinal muscles, occur let's see the pronunciation here.
reciprocally,
or together, while the chylomic fluid serves as a hydroskeleton by maintaining turgidity of the earthworm. So let's go over all these terms. This is invertebrates are the backbone, like worms. Just think of worms there. So the coleum, or the
chylome,
or coelom, chylome or coelom is the main body cavity in most animals and is positioned inside the body to surround and contain the digestive tract and other organs. So basically the main cavity inside. And the fluid inside the chylome is known as chylomic fluid. So that's that fluid inside there. It serves as a hydroskeleton. A hydrostatic skeleton or hydroskeleton is a flexible skeleton supported by fluid pressure. And turgidity. Yeah, turgidity is a state of being swollen or turgid, especially due to high fluid content. So here, if you look at the worm right here, so cross section of an oligogate worm. Let's see this.
oligocheat
worm, and the worm's body cavity surrounds the central typhlosul. Let's see the pronunciation. Typhlosul. All right. So let's take a look at how this setup is. So remember, it has no backbone. So what you're looking at is, yes, you have circular muscles all the way around. This is called the cuticle there. Metanephridium. This is nephridipore. Anyways, there's a longitudinal muscle layer going around the whole setup. It's longitudinal, so that means it's going to be going across like this. Yeah, so like that. And there's others set up here. There's the intestine over there, and there's the column of body cavity. This is a full opening there, and see the other stuff here. Ventral blood vessel, nerve cord, and other setups here. But anyways, basically you have these longitudinal muscles around, and you have this the chylome, the central cavity inside there, and there's intestine over there, which is quite interesting. Anyways, the body's cavity surrounds the central right here. This is in that red line there. Typhlosul. Actually, no, the center one's right here. Actually, this is the typhlosul. And yes, this one intestine is right here. And then the typhlosul. See the pronunciation again.
Typhlosul.
And then, yeah, that's that fold inside here, the tiflosole. So the tiflosole is an internal fold of the intestine or intestine inner wall. And yeah, the body cavity surrounds it. There's intestine and there's that inner fold of the intestine. Very, very interesting. All right, so let's just take a look at how the circular worms act there. Yeah, I'm not going to go over all those other random terms across this worm there. That's for another video if I go over worm biology. So many animals with a worm-like cylindrical body have a hydrostatic skeleton with a flexible skin and a water-filled cavity. They call the chylum. See the pronunciation?
Chylum or pseudocellum.
Yeah, chylum or pseudocellum or pseudochylum. All right. So you have the setup here. They move by peristalsis using opposite circular and longitudinal muscles, which act on the hydrostatic skeleton to change the body's shape. Yeah. So basically, you have the setup here. These contract, and then it changes the shape, and you can start moving over to this side. Contract, expand over here, or expand this side. Contract, expand backwards, and keeps going forwards. So longitudinal muscles, tail end, sense organs. Yeah, there's sense organs and mouth all the way on this side there, and the head end, tail end. And there's the gut inside or intestine. The circular muscles. Yeah. So you have circular muscles contract, and you have longitudinal muscles as well. Yeah, the contract interact such that you can bend the body and you can start moving. And yeah, so contract, relax, and move. And just take a look at this segment here of a simplified image showing earthworm movement via peristalsis. All right, so you have this worm. This contracts, and then what you end up having is this contracts, and then this starts this contracts and moves. So notice that this contracts, and notice how it projected itself. So contracts down, and then it can squeeze outwards, and now you make contact with this ground there, and then you start a compression side, and then you start a relaxation side. So this relaxes, it's over here, expands. So now you have a big area on the ground, and while this contracts there, so this is then you'll have a stretch zone, and then so you have these three set up right there, and then you keep doing this. So then these ones get bigger, while this contracts. So this and then this contracts over here, and so on. So now you have a whole wave of where these ones start relaxing, these ones contract, and now you end up with one block to the left. Yeah, so you get one block to the left there. Let's erase this. Let's draw that again. So you get one block like that. That's very, very fascinating. So a simplified image showing earthworm movement via peristalsis. Yeah, so that is how it moves. All right, let's continue further and look over here. So other animals such as mollusks, see the pronunciation again.
Mollusks.
Yeah, mollusks such as snails, octopus, etc. And nematodes, see the pronunciation, here's a type of worm.
Nematodes.
Yeah, nematodes, type of worm, possess, it's a mistake there on my part, possess obliquely or inclined striated banded muscles, which contain bands of thick and thin filaments that are arranged helically, like in a helix, rather than transversely, laterally, like in vertebrate, skeletal, or cardiac muscles. Yeah, so you have inclined, striated, contains bands of thick and thin, and are arranged helically rather than transversely or laterally or side to side. Yeah, it's very interesting. So advanced insects such as wasps, flies, bees, and beetles possess asynchronous muscles that constitute the flight muscles in these animals. These flight muscles are often called fibrillar muscles. See the pronunciation?
Fibrillar muscle.
Or fibrillar muscles, because they contain myofibrils that are thick and conspicuous or easily noticed. All right, so continue further. So MES note, asynchronous muscles are muscles in which there is no one-to-one relationship between electrical stimulation and mechanical contraction. These muscles are found in 75% of flying insects and have convergently evolved 7 to 10 times. Or yeah, convergently evolved 7 to 10 times.
Convergent.
Yeah, convergently. So unlike their synchronous counterparts that contract once per neural cycle, such as skeletal muscles, mechanical oscillations trigger force production in asynchronous muscles. Typically, the rate of mechanical contraction is an order of magnitude greater than electrical signals. Although they achieve greater force output and higher efficiency at high frequencies, they have limited applications because of their dependence on mechanical stretch. Yeah, so it's basically with these asynchronous muscles, when you input a signal inside, the force can still be continuous or just more than a one-to-one relationship. So it's not you input a signal and there's a force. You can input a signal and you can have multiple force. Similar to if you drop a basketball and it just bounces back and forth. So you have multiple signals. You can view that as a vibration versus if you just push the wall and then stop, you stop. Just to compare the difference between the types of synchronous and asynchronous muscles. But this is dealing with muscles itself, so we'll show you how it can basically have wings. For example, if insects fly based on just small input, and then the muscles themselves do the continuous vibration or stretching. Anyways, continue further. So we mentioned here of myofibrils, and also in the video above. So a myofibril, also known as a muscle fibril, or sarcostyle, let's just see the pronunciation here.
Sarcostyle.
Or sarcostyle is a basic rod-like organelle of a muscle cell. So here's an image here. This is skeletal muscle, and we'll compare it with the flight muscles soon, or the asynchronous muscles. Yeah, so basically what we have here is a skeletal muscle with myofibrils labeled on the upper right. So you have this part right here is called the sarcolemma, and then you have the nucleus stretch on the outside there. There's a mitochondria squeeze in between these myofibrils. So there's the myofibrils, those bundles right there. And these are other parts of it. If you zoom in on this side, what you get is this is called terminal cisterna, T-tubule over there. There's the triad, and there's that region triad, and then there's three. Notice those three of those. Yeah, three of those parts there. That's I think that's two of these terminal cisterna, and there's T-tubule, the pink one. Anyways, we're not gonna go over too much of that. And then this is the sarcoplasmic reticulum over here. Anyways, that's just parts of this this setup right here, this muscle cell. All right, continue further. So now we're gonna compare muscle fibers coordinating morphogenesis in a crowded space. Yes, so a lot of coordination is going to be needed for this morphogenesis. This is development of basically the whole body, the full body parts, etc. And let's take a look right here. So this is from ScienceDirect.com. So muscle fibers coordinating morphogenesis in a crowded space. Two examples of different mitochondrial myofibril architectures. Left is an adult Drosophila. Let's see the pronunciation.
Drosophila.
Or Drosophila is a species of flies and fibril flight muscles. Let's see that again, pronunciation. Fibrillar flight muscles containing round individual myofibrils in red, isolated from each other by large ellipsoid or just a 3D ellipse shaped mitochondria, shaped green. So here's the difference. So this setup's over here. So, this is the flight muscle, Drosophila. Drosophila. Anyways, there's the muscle fiber. Zoom in. There's the myofibril bundle, and notice the diameter is going to be bigger. Yeah, they are bigger and more easily noticed. This one has some small ones, irregularly shaped, etc. It's all over the place. Notice when this one is conspicuous. Just to see this term. So these flight muscles are often called fibrillar muscles because they contain myofibrils that are thick and conspicuous, easily noticed. Just to see, they're basically more distinguished over there, and then they so go over here. So flight muscles contain round individual myofibrils isolated from each other by large ellipsoid shaped mitochondria. So the mitochondria is in green and there. There's a cross section. You can see this in the cross section view over here, and this cross section from this angle. Notice how it's rectangle across there. So you have the mitochondria in green, the mitochondria. And also look at the sides there. There's the nuclei over there, stretched on the sides. So myofibrils, individual fibril type, so individual type like that one, each one. Yeah, so basically you can see the fibers together there. And then versus the mammalian skeletal muscle, we'll get that in a bit. So mitochondria squeezed by myofibrils, isolated and compact. So isolated, compact, and squeezed over there. And then if you look at the mammalian skeletal muscle, We'll go over that here. So right mammalian skeletal muscle contain bundles of vertically aligned cross-striated myofibrils with mitochondria concentrated in groups squeezed in between. These mitochondria form long, thin extensions along the sarcomeric. See the pronunciation?
Sarcomeric.
Or sarcomeric eye band. Eye bands, or I think it's an eye. Yeah. Let's see this.
Eye band.
Yes, it's eye, not an out. Eye bands that build networks. In both muscle types, the crowded cellular environment generates mechanical pressure that positions the nuclei at the periphery of myofibril bundles, adopting an elongated shapes. In both of them, you have you have over here the nuclei stretched outside, so it's being pushed outside of it over there. Yes, if you look at the mammalian skeletal muscle right here, so you have the muscle fiber bundle. Look at versus this one compared. There's a nucleus just on the outsides there. Then if you zoom into this fibril, or the myofibril, so then you get over here. So you get this setup here. So notice to compare this one. This one's just big round circles, round tubes, and it's pretty obvious. You can see them. This one is more just random shapes, and there's mitochondria squeezed in between. And then there's the cross-striated type. If you look at this across here, so this is the individual type, and this is a cross-striated. Basically, you have thicker ones, thinner ones, etc., and some other types of these banded myofibrils. Then mitochondria are highly networked, elongated, and branched. Yes, so the mitochondria, the highly networked, they're highly elongated. Yeah, branched out. You see them pushing outwards across here. It's like branches, etc. This one is just isolated in each one there. This one is squeezed and moved around, etc. Across the bands there. Let's erase this. And now if we continue further, let's look at another figure. This one's from Science Direct, but I believe it's a different article. But it's a similar article. So basically, I just want to compare it even further. You see, this is a muscle dimension, problem, structure, and dimensions of muscles in fly versus human. So top schematic representation of Drosophila. Drosophila and human muscles in a series of magnifications. Each muscle fiber contains hundreds of myofibrils that span the entire length of the myofiber. Sarcomeres are several orders of magnitude shorter, but must be assembled perfectly into a myofibril to connect both muscle tendon attachments at the fiber ends. In flying humans, sarcomeres are similar in length, so 3.2 micrometers, that's a millionth of a meter, in flight muscles, and 3 to 3.4 micrometers in relaxed human muscles. So yeah, so this time are similar, but flies very, very tiny. So it's relatively giant compared to the human. Yeah, the muscles, the flight muscles themselves. And so then, and many sarcomere proteins are well conserved. Yeah, similar length, and yeah, a lot of them are still well conserved in there. Yeah, so in both of them, they'll still have those similar proteins. And then, yeah, so that's the top. Let's take a look at the top right here. So, Drosophila flight muscle versus human skeletal muscle. So here is the length here. So muscle length 1 millimeter. And this one, muscle length 30 centimeters. So you have the muscle length, yeah, 30 centimeters is pretty big. And now you have this tendon there. And notice the tendon is much smaller, relative size. And now if you zoom in there, you get this, yeah, myofiber. They're all hundreds of myofibril. And then you zoom in. Yeah, so this one has zoom in there. And this length right here, this is taken actually from this thigh. So that's the thigh there, connected to the bone. And that's that long. This one's connected, this is this upper part of this fly for the wings. Yeah, so the flight muscles. Anyways, and you zoom in again, you get something that looks like this. This one is yeah, so they're more clear. You can see them. They're thick bands like that, more distinguished. And then myofibrils, and in both of them you zoom in. So this is a Z line, the end point, the middle, and then the Z at the other end. Sarcomere length 3.2 micrometers. And this one 3.0 to 3.4 micrometers. And you zoom in, and they both have similar setups like this. So it's very interesting. So the proteins are yeah, both well conserved. And so let's take a look at the bottom part. So bottom schematic of the sarcomere. Polar actin filaments, also called thin filaments, are anchored at the Z disc, or Z is Z dash green there. Yeah, or the Z label right here, and then Z disc. There's the green, so the green end. And then this is called alpha actinin. There's another protein, and these are anchored at the Z disc. So those are anchored over here. And let's see, this is the green ones right here. This type of protein anchored over there at the Z line there. The M line's in the middle there. And let's see what else is there. So thick filaments comprised of myosin bundles are centered at the M line, M and then blue. So that's the blue line right there. And then interact with actin with their myosin heads. So there's a myosin, there's the heads that we went over the video with. And yes, and then they connect to the actin over there. So there's the actin, those spirals over there. And there's the, at the end of here, there's other set of titinin, other proteins and so on. And yeah, this titin right here, this is the one that keeps it all centered in their movements. So this is centered in their movements there. And this is just zoomed out that you can see this more squiggly line at the end there. And we can scroll all the way back to our earlier setup, and just to show you that titinin over there. So if you scroll up, yeah, so there's the titinin or titin right there. And if you zoom out, yeah, so there's a titin, there's the myosin heads, and there's the actin over there. So it ensures that it's centered as it drags the actin closer in contraction. All right, so let's go all the way back here. Yes, all right. So that's the titin, and continue here. So titin, a connecting filament, is anchored at the Z disc and spans through the I band all the way to the M line. Yeah, so let's take a look at this. Yeah, so the titin goes from the I band all the way to the M line. So there's the I band right here. So there's the titin. Let me just erase all this. Yeah, so there's a titin from this I band, called the I band right there. Yeah, and then the M line is in the middle, the center right there. And this one right here is just this extension past here. And it goes all the way. Yeah, so the titin goes all the way across to the M line. And also from the other side. So yes, that is some very interesting stuff. All right, so now let's continue further. All right, so now let's take a look at this animation right here. This shows the asynchronous flight muscles and how they power flight in most insects. You have part A is the wings, it's blue with the black outline. Then you have B, the wing joints. So this is the wing joint right there, and this circular pink circles over there. And then the dorsoventral muscles power upstrokes. So these are the dorsoventral, and just a note here, MAS notes. So when you look at dorsoventral, the ventral is usually on this right side of the human there, or just the front side there, and then the back side, dorsal, posterior, or anterior. But if you look at animals or fish right here, the dorsal is the top part there of a typical animal there, and there's a dorsal up top there. So this could be the top there, and then the bottom is a ventral, but that just means the front. So this is the back, and this is the front. Yeah, so it's basically back and front. So the dorso means back, the top of it, and then the ventral means the front of this insect right there. So yes, just erase that. All right, and yeah, so let's take a look at here. So C is right here. This is this orange / beige color, and that's the dorsoventral muscles power upstroke. So what happens is, as this compresses, so as they compress via the sliding filament theory we went over, and then this compresses, what happens is these wings go up when that compresses. So notice what's compresses, the wings go up. So that's the power stroke. And then the dorsal-longitudinal muscles, they power down strokes. So when the dorsoventral is compressed, these ones are relaxed, so they just squeeze closer. But then when these ones compress, this animation should show more compression there. But anyways, when these compress, they expand outwards. And that pushes the dorsoventral, which are inactivated during this phase. So they're not compressed. And then they stretch outwards. So when this compresses, this stretches out upwards, which causes a downstroke. So as this stretches outwards, that pushes a downstroke. And notice how this mechanism right here, this vibratory / oscillation motion of these asynchronous muscles, shows that basically once you get it activated with some small neural input, then the muscles themselves go back and forth, and then they can move rapidly, right, like here. And hence why you'll have basically a much greater force output than the actual neural input, and it's due to the mechanical stretch of these muscles set up right there. It was very, very interesting.